Thank you so much for the opportunity to present my work here. First, let me do a brief introduction about lung cancer, which is the field where my lab works. There's two main types of lung cancer: non-small cell carcinoma, which is the majority of cases, and the most prevalent histology is called adenocarcinoma. And our main focus in the lab, which is small cell carcinomas, which is accounting for around 15% of cases. And as you can see here, lung cancer, non-small cell here, the first one, and small cell here, account for a really high numbers of cancer-related deaths per year in the US. And even though small cell lung cancer is a relatively rare subtype, it still accounts for a really high number of deaths per year, which is illustrating its aggressiveness. Very different to adenocarcinoma, where we have identified different molecular subsets, like for example, EGFR mutant patients or ALK fusion patients, and most importantly, identified targeted therapies for this subset. And this has really changed the way we treat patients and really have a really positive impact in the outcome of these patients. So unlike adenocarcinoma and small cell is pretty much like a black hole until very recently. And we don't really have any well-defined subset of patients that may benefit of a concrete therapy. And this is the reason why all small cell lung cancer cases are still treated with classic chemotherapy, which is very toxic and not the most efficacious. Even though most of the patients respond pretty well, they will rapidly develop resistance. So this is a problem because in the end, that will derive in really low survival percentages for these cases. You can see that for extensive stage disease, which is most of the small cell cases, the 5-year survival is around below 1%. So we have recently defined different molecular subtypes of small cell lung cancer by performing molecular characterizations on small cell lung cancer clinical samples. And we have identified 4 main subtypes, which are defined by the dominant expression of 1 out of 4 transcription factors, ASL1, ALD1, PAL2A3, or JAP1. And this is important because it's the start of defining subset of patients that each of which have or show differential expression of genes that may be therapeutic vulnerabilities, and those may respond differently to a concrete targeted therapy similar to what we see in adenocarcinoma. But we're not there yet. The main cause of, in general, cancer-related deaths, and particularly for small cell lung cancer, is metastasis. So you see that we get metastasis to the lung, to the brain, to the liver, and this is what ends up killing the patient. One of the issues about studying metastasis in small cell lung cancer is that we don't really have great models that really reproduce the disease progression. And you can say, okay, why don't you use those like cohorts that have been molecularly characterized? We cannot really do that because all of these tumors that have been RNA-seq, they really come from early stage resected tumors, because this is how we mostly get clinical samples from the clinics. Only those tumors that are removed, which are the early stage, we can work with generally. So these don't really represent the whole progression of the disease. Also, like all of the characterizations that are available so far are only bulk. And as you may know, these are not the most informative, because basically in bulk analysis, what we're seeing is that even though one concrete system may have different clones, in the end, when we do bulk analysis, we get expression or information about the average clone. So we're missing a lot of other clones that may be also interesting or driving some phenotypes. In single cell RNA-seq analysis, we can get information for all of those clones individually that may, which may be more informative, because some of these clones may be driving an important phenotype. So just as a brief conclusions of this introduction, small cell lung cancer are really bad, and metastasis is what causes death in this setting. It's very hard to get small cell clinical samples that represent the whole metastatic process, because we usually only get resections with an early stage. And the data that is available for this is usually bulk data, not single cell. We have identified potential subtypes of small cell lung cancer, but which may have clinical implications. But even so, we don't really have still targeted therapies that will benefit any of those subsets. So they are still treated with classic chemotherapy, which is not the best option. And these two problems, so to say, or unmet needs, are the ones that I'll try to address in this presentation. One of them, how to identify novel mechanism of metastasis in small cell lung cancer. And the second one, novel therapeutic targets for these tumors. So for the first one, as I said, what has been characterized so far are resections, which are like the samples that we get more often. but these only represent early stages of the disease. So we had to optimize a protocol to process samples for single-cell RNA-seq that will represent different stages like local metastasis and distant metastasis. So we optimized the protocol to process not only resection for single-cell RNA-seq, but also biopsy, coordinated biopsies, pleural effusions, which are liquid that get originated in the lungs of cancer patients, and they need to get drained because it causes a lot of discomfort. And that liquid actually contains a lot of tumor cells, so it's very informative. And as well, we have optimized a protocol to process fine needle aspirations, which are like very tiny biopsies. So the idea of this is expand the type of samples that we can perform sequencing on in order to capture the whole metastatic process in its whole extension. So the protocol is very straightforward, nothing magical. We get the resections or the biopsies, basically the solid tissue, and we cut it into small pieces, and we proceed to a small or a short mechanical and enzymatical dissociation. For the fluids, the pleural effusions that was mentioning before, we do a centrifugation by gradient in order to get the cells included in the liquid. And then we get to a point where we have a solution with red blood cells that we want to eliminate, and then we stain the cells basically to solve for viability, and we can do additional stainings if necessary, like for example, CD45 stainings, we are interested in immune cells, and I will talk about that in a minute. And then the point is to sort the viable cells, because single-cell RNA-seq requires high viability. And we are very close to publish this protocol in the SAS protocol from Cell Press journals, in case you are curious about the details of this, or you can just contact me and I'm happy to share it. So there's two ways that we can sort the living cells here. We can just sort for living cells in general, but we can also stain for CD45 that will stain the immune cells. In that case, we can have the non-immune cells, which will be enriched in tumor cells, and then the immune cells. And why do we do this? For small cell lung cancer, we don't really need it, because you can see here that these tumors are not really very infiltrated by tumor cells. This is a MIBI image, which is like a imaging technique that stains for different markers at the same time. These are all markers of tumor cells. And here to the side, we see markers of immune cells. And as you can see, it's like pretty much completely black. For adenocarcinoma, that's different though. Here we're showing markers for tumor cells. And when we stain for immune cells, we see a lot of cells here. So in adenocarcinoma, there's a lot of immune infiltration. Sometimes tumor cells may represent less than 5% of the cells. And if we are interested in the tumor cells, maybe we don't want to sequence those many immune cells. So the idea of sorting immune and non-immune cells would be to get them both separated. And if you are interested, if you are an immunologist and you're interested in immune cells, you can sequence only the immune cells. If you are a tumor researcher and you're only interested in the non-immune cells, you can go for that. But you can also mix both at the decay ratio. So you have the representation that you want for each of the cells. And here I'm showing an example of a clinical sample. At the beginning, we were staining only with DAPI for viability, which was not optimal. You can see here, seeing the discrimination as usual. And then we try to be inclusive here with the DAPI. And here we are separating by CD45 positive and negative, probably not the best gating, but still. And it was challenging because we were not able to distinguish between those cells that were negative for DAPI and those that were small debris that will stain negative for DAPI like this. So that's why we decided to include a second viability marker, which was calcium. Probably not the ideal either, but most informative for sure. And here I'm showing you another clinical sample. In this case, it's an adenocarcinoma with low tumor content. Again, after the single discrimination, we will stain and select for the negative for DAPI. And then here you can see that with the physics, we have like a second dimension for viability, which really helped us distinguish which cells were actually alive. And if you notice here, what I was mentioning before is P5, which will be the tumor cells, are only like 3% of the whole cell. So that's why if you're interested in tumor cells and you sequence these samples like that, only by sorting the viable cells, you will get only 3% of tumor cells or non-immune cells. So if you sequence around 3,000 cells per single cell RNA-seq run, you may end up with only like 90 tumor cells, might be optimal for analysis. And you can see here another adenocarcinoma sample. This is how most of our samples look like. If you start with a very small biopsy, you don't get many cells to work with. These are the cells that we run to kind of set up the gates. Completely not optimal for sure, and for sure, no controls at all, which is something that the flow cytometric people were shouting me about, but we don't really have cells to waste in that case. And what we will always try is to be very inclusive. You can see here, probably we are sorting a lot of debris, especially because our P1 was also very inclusive. And then in this case, we were not able to even sort the CD45 positive and negative into different tubes because there were so little cells that we need all of them together to accumulate the minimal mass that we need to actually proceed for sequencing. So really not straightforward, what we were able to kind of figure it out and do things analysis on these samples. Some of the issues that we ran into is for some samples, we had inefficient separation of live and dead cells by calcine. That is because when sometimes you have a sample that has a lot of cells, you may not add the right amount of calcine, or you might not able to incubate with calcine as much as you wanted, because in the end you want to sort the cells, you need to bring them to the core after, and coordinating all of that may be ahead with clinical samples that may arrive 2 hours later than what they should. So one of the ways that we had to fix it was to add always excess of calcine. Another issue that we run into is inefficient separation of immune and non-immune cells. And this is probably the same in most cases because you start with a sample with a lot of cells, so you probably don't add enough CD45 antibody. So adding extra excess usually helps improving the separation. And now we're gonna try something that I've never done before, so I hope it works. This is one clinical sample that we actually sorted. Again, you can see here, we got the single discrimination. We tried to be inclusive with the DAPI, and we got this. So I wanted to ask you guys which of these three options you think is correct. And to do that, we're using a system that is called Poll Everywhere. So if you can scan this QR code with your phone or just type this, just type this address in your browser, you can access to the poll and you can vote which one you think is the correct one. And you guys have 1 minute to do that. Okay, it's working. Okay. I think it's not changing because everyone agrees on the same. Okay. Oop, okay. Okay, so what related to their answer? Okay, so most people are right. I would say the right option is B. This is the on the x-axis we have FITC, calcine, and on the y-axis we have CD45. So if you, where's the pointer? Okay, here. So the FITC negative cells will be calcine negative cells, will be the dead cells, and then the FITC positive cells that are CD45 negative will be the tumor enriched cells, and then the CD45 positive will be the immune cells. So this one was very easy. I know you guys are very smart, but this one may not be as easy. Again, we have the single discrimination. We were very inclusive on P1, and we were also very inclusive in DAPI because the sample was looking a little odd. And we got all these populations, all these like smear or staining levels. So if you guys have these three options, in this case, FITC is on the y-axis and CD45 is on the x-axis. Which one would you say would be? And Michael, if you can clear this one before we start. You need to put your mouse here, down here. Okay. And clear this also. Thank you. Okay, now we can start. If you can take the mouse off there so we can see the, oops. Okay. Okay, so people have different opinions here. This one is not as easy, right? So this is what we had to face every single time we would sort a clinical sample because all of them look different. It was very challenging, and without the help of the MSK flow cytometry core, this wouldn't have been possible at all because I had no clue about what would be immune cells or whatnot. Okay, so we have different opinions here. To be honest, I'm not entirely sure what would be the right answer, or it keeps on changing. Okay, cool. So I would say this is probably the right answer. There's like this population here that may be a little more negative for CD45, and then all these populations that may represent the different immune populations there. So I would say that's the one. But again, to be honest, this team was like guessing every single time, try to do our best. Okay. So somehow we managed to produce a small cell single cell atlas. I don't know how that was possible. Thanks to a lot of patients from the flow cytometry core in MSK. You can see we had, if we zoom to the epithelial cells, we have all these subtypes, and if we focus in the small cell tumor cells, we have these 21 human tumor specimens that belong to different treatment categories. Some of them were untreated, some of them were treated with chemotherapy or chemoimmunotherapy. There was a high heterogeneity of samples and also a high heterogeneity of size. Some of them were from the lung, primary lung tumors. Some of them were from the lymph nodes. Some of them from liver metastases, etc. And one of the things we noticed is that small cell tumors show really high heterogeneity. And you can see here, this Shannon entropy is anti-correlating to the heterogeneity. So heterogeneity is higher when lower the Shannon entropy. And you can see that small cell is among the lowest, even smaller than adenocarcinoma, and of course, smaller than the immune cells, which should be very similar interpatient, and that seems to be the case. So we were surprised to find this much heterogeneity in small cell tumors because they actually seem pretty homogeneous when you look at them at the histological level. But this may be underlying the reason that why they are so aggressive and why they adapt to treatment and they become resistant so easily because of this richness in heterogeneity. And another thing we noticed is that we were able to detect 3 of the 4 subtypes in our cells. You can see small cell cancer A, P, and N. But when we made this weird plot where we assign to each of the cells in each of the tumors a probability to belong to one of the 3 subtypes, you can see that most of the cells will like group within one of the corners, which will mean that those cells will look like very much like one of the subtypes. But you see that there's a lot of cells that fall in the middle between the 2 subtypes, which kind of suggests that there might be some subtype plasticity. There might be some interconvention from one subtype to another one in response to therapy or during progression of the disease. So we were very, very surprised to find that. This makes the stratification of patients a little more challenging because it seems that subtypes are not really static or rigid, but rather plastic and interconverting. And one very cool thing that we found was a very small cluster. These are all the small cell tumor cells. We found this small cluster that was pretty surprising because it showed a high subtype uncertainty, meaning that it was very stem-like or very differentiated. And if you see here, this plot down here is showing each of the colors is a different sample. So this graph is telling us that this cluster 22 was integrated by most of the samples, including in our cohort. So all of the samples had cells that belong to this cluster. So you think that something really universal was going on with this cluster. And when we had a look at which programs were enriched in that cluster, we were surprised to find that they were enriched in metastatic programs. So you think that this cluster may be driving metastasis somehow. And one of the characteristics of the cluster was that it was very highly expressed in PLCE2, a gene that is located in the membrane, and very highly expressed in this cluster compared to the other clusters that we found. And when we had a look at the expression of this PLCE2 in different sites, you can see that expression of PLCE2 was higher in any other site compared to the lung, suggesting that expression of PLCE2 may get enriched during the metastatic process because it's the lowest in the lung, and then it gets increased in the different metastatic sites. So in order to test whether PLCE2 was actually driving metastasis, what we did was different experiments that I'm going to talk about. But we did one of them that was like very key, which was intracardiac injections. Basically, you inject the cells in the heart of the mouse, and then they get distributed all over their body. And if they're able to colonize an area and produce a metastasis, you will see that by luminescence. You can see here that for one cell line for which we generated two isogenic cell lines, one control with no PLCD2 expression and one overexpressing PLCD2, you see that PLCD2 overexpression increases the number of metastases generated. And the opposite happens if we knock out PLCD2 in a cell line that expresses high levels of PLCD2. You see here that there's a lot of metastases here, and the number of metastases gets reduced when you knock out PLCD2. And when we had a look at how this cluster 22 that we were talking about may be ready to survival in these patients, we saw that those tumors that had a higher percentage of this cluster in them would do much worse in terms of metastasis, in terms of survival, sorry. And this was single-center-seq data, but when we try to detect this PSC2 high cluster at the protein level by performing this multiplex immunohistochemistry, here you can see an image of one tumor, and you can see in pink all of the PLC2 positive cells. We were able to predict the same. Again, the cluster will predict in an independent cohort very poorer survival in those tumors with high representation of this cluster. So to summarize this part, we have established protocols to process a variety of clinical specimens for single cell techniques. We have made available single cell data from a cohort of small cell lung cancer specimens that may represent the whole metastatic process. And we have identified one novel mechanism of metastasis driven by this small population that seems to be expressing high levels of PLCD2. And we have also identified this plasticity between subtypes that I was mentioning before. So part two, as I mentioned before, most of these small cell lung cancer tumors, independently of the subtype, independently of anything, they will be treated with chemotherapy in every case. And even though they will respond usually, they will develop resistance very quickly. So in order to kind of maximize the response to chemotherapy, what we do was to run a CRISPR screen where we would try to identify therapeutic targets that were sensitized to cisplatin. And to do that, we took different small cell lung cancer cell lines, and we leverage a druggable genome library, which will contain only hits that will be pharmacologically targetable. And we ran in parallel untreated and chemotherapy-treated condition, basically to compare them both and select those hits that were specific for the cisplatin treatment condition. And in this cell line, in this screen, we included a bunch of cell lines from different subtypes and showing differential sensitivity to chemotherapy. And we found that what hit XPO1 was present in most of the cell lines. So we were interested in this hit. We didn't know anything about it before. And it turned out to be a nuclear exporter that is in charge of exporting mRNAs and proteins from the nucleus to the cytosol. And many of these mRNAs and proteins are actually tumor suppressors and oncogenes. And that's why this target has been gaining a lot of attention in cancer research. And many inhibitors have been developed that are potent and pretty safe. And some of them are even approved for their use in certain settings, like hematological malignancies. So these targets seem to be pretty interesting because basically there's an inhibitor available in the clinics. So that will allow any findings that we will make to be very easily translated into the clinics. So before doing anything else, we wanted to do some functional validation in vivo, and I'll tell you about some techniques that we use to do that. So we wanted to do something cool with this. So we tried to do this functional genomics, but not using simple models like cell lines or organoids, but using PDXs, which are patient-derived xenografts. To those of you who are not familiar to this, basically you take the tumor from the patient, you put it into immunocompromised mice, and the tumor grows in the mice forever. So you can use those tumors that grow in mice to test different therapeutic approaches or to do functional genomics, like in this case. And to do the functional genomics in PDXs, we needed to generate Cas9 expressing PDXs. And to do that, we generated this construct that basically expresses the constitutively CD4, a short form of CD4 that would allow for selection of those cells that have incorporated the construct, and that has a conditional promoter for Cas9 that will be activated by doxycycline. So basically, this construct will express CD4 in any case, but will be off for Cas9 when not under doxycycline. And this will allow the selection by CD4 for those cells that get the construct. And we saw that when you activate this promoter with doxycycline, you not only get expression of Cas9, but also you get higher expression of CD4. Expression of CD4, even if constitutive, also gets enhanced. And it's going to be very interesting for later in the talk. The way we get this PDX is basically we collect the tumor from the mouse, we dissociate it into single cells, and we perform a very quick spin transduction with very high concentrated virus. And this way we're able to transduce at least part of the cells within those tumors. And these cells will be like re-grafted immediately. So in the end, the time that the cells are outside the mouse is only about 1-2 hours to make sure that the whole process is done in vivo and there's no in vitro part. Then when those tumors that have been transduced, that have been infected with the virus, with the construct grow, we can collect them again, dissociate them again, and we can select for those cells that have the construct with the CD4 selection marker. And we can do that by flow cytometry or by magnetic bake separation. And then those tumors or those cells can be cryopreserved and revived or can be used to run functional genomics. So with those cells that have been transduced that express CD4 and that may express Cas9, we initially made some tests to see how the system would work. You can see here that with no doxycycline, those cells would express CD4. And we tried to make a test by transducing these cells with GFP. You see that the cells express GFP and see how we would be able to knock out GFP in these mice. You see, we were very surprised to find that when we induce by doxycycline, not all of the cells get induction of CD4. Only some of the cells will get induction of CD4, while some of them will remain positive, but not like super positive. And when we have a look at what's going on here, if we gate it for the cells that are positive but not induced, you see that the expression of GFP is maintained. And if you have a look only at the cells that get CD4 induced, you see there's a reduction of GFP. So this is very interesting because it was suggesting that on the cells that express CD4 but not get enhanced expression of CD4, we don't really get Cas9 expression. But the cells that get enhanced CD4 expression get high expression of Cas9, and that's where the editing actually happens. So we have a way to basically monitor which cells within one tumor are able to edit and which ones are not. And how do we do functional genomics on this? One example would be clonal competition assays. I'm not sure if you guys are familiar to this, so I'm going to go very quickly over this. So let's say that we have a Cas9 expressing cell line, and we infect it with a sgRNA targeting a gene that is not essential or involved in proliferation. And we couple to that sgRNA or to that knockout a fluorescent protein like GFP. In parallel, we do the same, but with a control non-targeting sgRNA that will edit nothing, and with a different fluorophore, like for example, N-cherry. So if we mix these two populations together, and again, gene X, the gene that we are knocking out, is not essential or involved in proliferation, you will see that over time, both of the populations will grow at the same manner. So the relative abundance of each of the populations will remain stable over time. But what happens if we do the same, but in the case where gene X, the gene that we are knocking out, is actually essential or involved in proliferation? What do you think may happen? Michael, can you clean again? Okay. Okay, let me go. Okay. I think you need to take the mouse off the. Okay, what's going on? You can continue answering. This would not affect the questions. Okay. So it seems that most people think is the option C, and that's correct. So what will happen will be that as gene X, the gene that we are knocking out is essential, over time, the population of GFP positive cells will deplete, and we'll see an enrichment of the control and cherry positive cells and a depletion of the GFP positive cells. So what happens in the PDXs? So pretty much the same. Let's say that we have, again, we are knocking out a gene that is essential with GFP, And then we have a control N cherry that will not edit anything. So we have our mixture of cells, and then if there's no Cas9 editing, both populations will again grow the same, and the population abundances will remain stable. But in those cells in which Cas9 is editing, there will be knockout gene X, and then those cells will deplete over time. So in the PDX, we don't get the whole time course, but we get only the final snapshot. But that is enough to interpret the data, because if you see here, even in the same tumor, we will have a population of cells where there's no Cas9, and we'll see that the populations remain the same. And then in those cells in which Cas9 is highly expressed, we will see that there's a depletion of the gene X knockout cells. So this is a way to kind of validate that a gene might be essential in vivo in a tumor that resembles really the behavior of a human tumor. And this is how it actually looks by flow cytometry. You see, we are doing the single discrimination again and trying to get only those that are DAPI negative. Here you see for CD4, we have this CD4 positive and then this CD4 induced, which are the ones that express Cas9. And you can see that if you gate on only the positive ones, you see that the population of cells in this case that have GFP will be around 76%. The control population with NCH will be 10%. But if you get specifically on those that get induced CD4, which also get induced Cas9, you'll see that there will be a depletion of the gene X GFP cells and an enrichment of the control cells. So with one snapshot in the end, you can really get information about whether one gene is essential or not. And this is what we do. We go back to the project that I was talking about like 10 minutes ago. We identify XPO1 as a target to sensitize to cisplatin. So we wanted to validate that in vivo. So what we did was to do this clonal competition assays, but it was a little more complicated because we have also the variable of chemotherapy treatment, cisplatin. You can see here that when there's no Cas9 editing at all, you see that the populations treated with cisplatin don't really change. So they remain the same. However, when we don't do any cisplatin or any chemotherapy, you see that those cells with the XPO1 knockout will deplete a little bit. And this is because XPO1 is actually an essential gene. But the most interesting is here. When we treat all tumors with chemotherapy, you see that in the non-Cas9 editing condition, there will be a similar abundance of both populations. But then on those tumors treated with chemotherapy, we see a very high depletion of the XPO1 knockout genes. So this kind of suggests that when you knock out XPO1, the cells become more sensitive to cisplatin. So then we went forward to study a little more about exporting one in small cell lung cancer, and we saw that it was actually more highly expressed in small cell lung cancer than in any other tumor type, including hematological malignancies, which is the setting where exporting one inhibitors have been approved. And we were able to validate this at the protein level by performing immunohistochemistry in clinical sample TMAs. You can see again that exporting one is not only more highly expressed in small cell than in other lung tumors, but also that is ubiquitously highly expressed in all small cell lung cancer tumors, suggesting that the target is there. And when we treated different patient-derived xenografts with the combination of cisplatin and selinexor, we saw a really high response. You can see here two different models belonging to two different small cell lung cancer subtypes. You can see that the response of these models to the combination of cisplatin and etoposide in clear blue is very low. This is the therapy that patients will actually receive in the clinic. But when we combine cisplatin with selinexor, the response is very, very dramatic compared to the standard of care, suggesting that this combination may be an interesting therapeutic approach for small cell lung cancer tumors. And when we try to find out a little bit about the mechanism, we perform RNA-seq on different cell lines and PDX is treated with selinexor, the inhibitor exporting one. And we found that there were only 14 pathways in common in all of the models, and few of them were related to the AKT-mTOR pathway. What we saw is that when we treat small cell lung cancer cell lines with chemotherapy, they get induction of the AKT-mTOR therapy, potentially as a cytoprotective or anti-apoptotic mechanism. However, when we treat them in combination with selinexor, with the exporting one inhibitor, we don't see this anymore. So this was suggesting that the AKT pathway may be related to chemotherapy resistance. And what we did to kind of test that is to overactivate the AKT-mTOR pathway in different small cell lung cancer cell lines. And you see that when that happens in the pink curve, you see that they are less responsive to chemotherapy. So we came up with a model where small cell lung cancer may be able to activate the AKT-mTOR pathway as a response to chemotherapy. And when you also inhibit exporting one, this doesn't happen anymore, and that makes these tumors more sensitive to chemotherapy. So take-home messages of this part is that exporting one is very highly expressed in small cell lung cancer, so that suggests that it's an interesting therapeutic target for pretty much every small cell tumor. When we inhibit exporting one, this strongly sensitizes small cell lung cancer tumors to chemotherapy, and the response is way better than the current standard of care. And that this mechanism seems to be mediated by the impairment of AKT-mTOR signaling that happens after treatment with chemotherapy. And to that, I would like to thank all my lab members, collaborators, and if you are curious about the details of these projects that I presented, here's the publication, so you can reach out to me. And particularly would like to give a very special thanks to the flow cytometry core that has been not only amazing, but particularly very, very patient to me and making it always possible to sort these samples, even though I would be like 2 hours late, no controls, few cells, and particularly during the COVID time. So I really, really, really would like to thank you all, guys. You're amazing. And happy to take any questions.
Thank you, Alvaro, for this great talk. Are there any questions in the room?
It's a really great talk. I had a question about the atlas actually. So you guys are basically separating out this population of epithelial cells. I was wondering if you can go in and distinguish malignantly transformed epithelial cells from potential non-neoplastic contaminants using like copy number, aneuploidies, or any mutations you can detect in
Right.
RNA-seq.
So for the small cells, that was very easy because these cells have like a lot of genomic aberrations. For the adenocarcinoma cells that we're using as comparator, that was a little more challenging because these cells really resemble lung epithelial cells. But we had like two ways of kind of distinguish that. One of them is sequence actual normal human epithelial cells and see how they will group with them to kind of distinguish the ones that are tumor, but also by checking some previously known copy number aberrations, we will see, even though it was not as sensitive as a small cell, that will give us a hint of what would be actual tumor.
Do you have any idea why the doxycycline enhanced the CD4 expression in the RTTA construct?
We have no idea. I
Magic.
guess we are like, in the end, they're very close in the construction, and that might be the reason why we don't know. When we noticed about that, we were very surprised and kind of disappointed, but then we tried to actually get advantage of that.
All right. Thank you so much, Alvaro.